Greetings again, Biology 101 fans. We here at North Shore Community College bring you yet another episode in your lab videos, this time from an undisclosed location. Though this one's really not a lab video in a sense because we're just going to be constructing a model of DNA here. I've got this model kit. This has got all the pieces that we can put together and form a model of DNA. When we get through, the model of DNA should look like this picture here. This is showing it on the stand. We don't necessarily have to use the stand, but it will look like the double helix that you've got to know and love already. Before we get started with the model kit, let me show you an illustration of DNA just for the sake of orientation. This is a pretty good illustration of DNA here. This is the famed double helix that you've been seeing for quite a while now. And it consists really of, you can think of this as really in two portions. There's this ribbon here called a backbone, and the second ribbon here, second backbone, double-stranded. That's where the term double-stranded comes from. And the backbone consists of a sugar and a phosphate, sugar and a phosphate. These are alternating, so often this is called a sugar-phosphate backbone, or as written here, phosphate deoxyribose. That's the sugar backbone. So here's the backbone. Here's a backbone. So that's one part. And in the middle of this is the base pairing. This is the base cytosine hydrogen bonded to the base guanine. And it's always going to be a GC or CG base pair. Adenine will always pair with thymine and so forth, with two hydrogen bonds between those. So you get the base pairing in the middle, the backbones on the outside. So think of the DNA that way, at least for the double-stranded molecules. Backbone outside, base pairing on the inside. In this particular model, there are sugars represented by these reddish orangey pentagons with a couple of pegs on them. The phosphate is this light purple or lavender diamond-shaped thing, one peg, and then the bases are these flat pieces, A for adenine, G for guanine, T for thymine, and C for cytosine. Now I've labeled these just to make it easier in the beginning here, but there is a color coding pattern if you haven't figured that out yet for the bases. The color coding pattern goes like this. Again, thymine is orange, so I label it as T, so it will be easier to begin with. Adenine is blue. Cytosine is yellow. And guanine is green. Right, so remember, again, these base pair in a specific way. Thymine always pairs up with adenine in the middle. And again, it's held by two hydrogen bonds. That's represented by these two pegs. Cytosine and guanine, by the same token, will pair up exactly specifically, only these two together. And these form three hydrogen bonds, again represented by these three pegs fitting together. So GC base pair, AT base pair. But the base pairing comes at the end here, not the beginning. But that's the color coding that the manufacturer picked for this particular model. So what we're going to do is put things together piece by piece to build a model of DNA. But DNA is synthesized from what's called nucleotides. And a nucleotide is a sugar, a phosphate, and a base. Here's a phosphate, here's a sugar, here's a base. Before I put these together, let me orient you to the sugar a little bit. We got to look at the sugar anatomy closely. So I've labeled this one more specifically, S for the sugar, but then you see the 5 here, and that's really representing this peg. There's a peg here with a little bit of a ball on the end, and that is called the 5' carbon. That's what's represented on the deoxyribose sugar. There's a peg on this side here, but there isn't any little ball here. That's going to fit the base. That's the 1' carbon. There's a little hole in this by this corner. That's the 3' carbon. So the numbering of the carbons in the ring goes 1, 2, 3, 4 for this corner here. 5 is going to be this. I had to write it on the surface because you can't write on here to make it readable. Right? So that's the 5' carbon. 3' carbon represented by the hole. 1' here. Just to clue you in for a moment, the sugar and the bases both are ring structures. So this has a system of numbering to designate positions on the sugar. The rings of the bases also have positions designated by numbers, and so you distinguish the sugar bases, sugar numbering by primes, and that's the only reason why I call it a 5 prime. But what I want to do is build a 5 prime nucleotide first. So to do that, I'm going to take a phosphate and stick it on the 5 prime carbon. And I have to stick the phosphate on in this particular orientation for this model. It's just a peculiarity of this particular model that I have to put it on in this orientation. But notice the phosphate is on the 5 prime carbon. So that's 5 prime phosphate on the deoxyribose of the sugar. Then a base, any base I want, will go onto this position, onto the number 1. And I happen to pick cytosine. So cytosine, the base is on the number 1 carbon, the phosphate is on the number 5 carbon. And that's what you call a 5 prime nucleotide. The reason I'm making a 5 prime nucleotide is because that's the structure of the nucleotides that cells use to synthesize DNA. They always come from 5 prime nucleotides. To remind you of some terminology, or to introduce some terminology, here is it written out in a little bit more detail with the terms. So base, 1 of 4 here. Here's a base. Here's a sugar. Here's a phosphate. Right? Base, sugar, phosphate is a nucleotide. Phosphate goes on the 5 prime again. Base onto the 1 prime. That's a nucleotide. So what I'm going to do is I'm going to construct 5 prime nucleotides out of all these pieces first. And then I'm going to construct the model of DNA from all the 5 prime nucleotides. It has to be all 5 prime nucleotides. And there's a little peculiarity here. If in this model, the plane of the base has to be perpendicular to the plane of the sugar. The sugar these are both relatively planar. Certainly the base in DNA molecules, and the real DNA molecules, is a planar structure. The sugar is kind of planar. Whether they are really perpendicular in reality in the DNA is a question we don't have to really deal with. But I have to put the phosphate on this position with the edge, the free edge in a sense, without the peg or the hole on the outside of the structure. But I'm going to make all the nucleotides out of a base, sugar, phosphate, and then I'll be back to show you how to put the nucleotides together to form a DNA strand. So we're back. I've got all the nucleotides assembled now. Here again was the first one that I did. Cytosine, base onto the sugar, phosphate. Phosphate on the 5 prime, base onto the 1 prime. Now there's another peculiarity of this particular model kit. There's nothing to do with DNA, just this model kit. You see a straight side on all of these bases. Straight side with the pegs. I have to point these in exactly the same orientation for every nucleotide, just as the model, these things fit together and make a nice DNA model. So here's the straight side, and I'm pointing it away from this face of the sugar. I could point it like this, and that would work if I have all the nucleotides exactly oriented the same way. I just happen to construct this one in this way, and that means all the others have to be oriented in exactly the same way, because then they're going to fit together correctly. So notice this same orientation. 1 prime with the base, 5 prime with the phosphate, and the straight edge pointing away. 1 prime with the base, 5 prime with the phosphate, straight edge pointing away. So now that all the nucleotides are assembled, though, now I can join the nucleotides together and make bigger molecules. So if I take, now when you join the nucleotides together, they have to go together in a particular way. This is really how it happens in cells. It's specificity for the enzymes. That's just the way it goes. So here's a second nucleotide, sugar phosphate. I'm using adenine here because I felt like it. I could use any other base. but I'm going to take this phosphate and join it to the 3' carbon of this nucleotide. So that is phosphate from this nucleotide fits onto the 3' nucleotide of another sugar, of another nucleotide. And then you have two nucleotides stuck together. So just to clue you in on a little bit more terminology, if we take two nucleotides, join them together, we get a dinucleotide. Di meaning two. If I put a third nucleotide on there, 3 nucleotides joined together in a short chain is then called a trinucleotide, tri meaning 3. And if I take a lot of nucleotides, join them together, I'll get a polynucleotide chain, poly meaning many. And a polynucleotide chain is also called a strand, and that would be called a strand of DNA. Or it could be RNA too if you use the RNA bases and you use ribose as the sugar. Same thing, a polynucleotide chain. And this happens to be DNA because we're using deoxyribose as the sugar and we're using thymine as one of the bases. And we're making DNA here. There is certainly models of RNA you can make too. Same thing though, here's a dinucleotide. I'm going to take another nucleotide. I'm going to take the phosphate here on this single nucleotide, join it to this dinucleotide by hitting again this 3' carbon on the sugar. So that's joining that phosphate onto the 3'. That's always the way the synthesis goes in DNA or RNA for that matter. So the synthesis is going from what's called the 5' end to the 3' end. So these chains it's called asymmetrical. There's a particular polarity, as it's called, or orientation to the change. Here is the 5' end, that's up. Here is the 3' end pointing down. Right, 5' to 3'. And it's synthesized always 5' to 3'. So whenever I'm going to join another nucleotide, I put it onto the 3' carbon. So there's 4 nucleotides joined together. I do another one onto the 3' carbon, 5 nucleotides into a chain. Right? And notice they're always, the bases again are always oriented in the same direction. I'm going to continue this on to make 12 nucleotides in a chain, and that's going to be the limit for this model. I'm going to make a polynucleotide chain out of 12 of these nucleotides. So I'll be back when I've got that finished, because otherwise you're just going to watch me do the same thing again and again. But then the crucial part happens when I actually have to make the second strand and do some base pairing. So I'll be back in a second to show you the completed one side of this chain and then start the base pairing. So we're back after completing the first polynucleotide chain. Here it is. So here's the nucleotide that I started with, the cytosine up on the top. That's the 5' end, and there's 12 of these. Just to convince you, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 nucleotides. We'll call this a small polynucleotide chain. Now the next thing to do, of course, is to make the complementary strand, a complementary polynucleotide. So this is going to take me swapping this around, because what happens is here's a thymine here. I'm going to pair that up with a cytosine in the orange. Sorry, pair that up with a thymine in the orange, adenine, thymine. And when I do that, here I'm going to base pair it first. Then for the next one, we got a thymine. I'm going to put an adenine on it. But really what happens is this is going to be synthesized in the same manner as the first strand that I made here. Phosphate here is going to be bonded onto the 3' carbon of that sugar. And then you base pair. Now what happens is in biology, in cells, there's an enzyme called DNA polymerase. And DNA polymerase will read the base pairs, match the bases, and then connect the phosphate to the adjoining nucleotide that's already base paired here. So same process, phosphate here goes on to a 3' carbon of that sugar, and then you base pair the cytosine with the guanine, and you get a base pair. That's going to be synthesizing then. Again, this is going to be the 5' end, 5' to 3'. So you're still synthesizing 5' to 3'. This just happens to be the way the structure of the nucleotides is made. Is you have what's called nucleotide triphosphates, and the triphosphate is always on the 5' carbon as an ATP. ATP is a ribotriphosphate nucleotide. Same thing, phosphate onto the 3' carbon, base pair, in this case the guanine to the cytosine. And again, in cells, this is going to happen because there's an enzyme that matches the bases and then joins the phosphates to the sugar. Thymine here requires another adenine, phosphate onto the 3' carbon, bases pair up. Cytosine here requires a guanine, phosphate into the 3' carbon, bases pair up. All right, so it just continues till you get to the whole sequence here, the whole double-stranded molecule. And so I'll be back when I've got that done, because I think you've got the point by now, right? So here is our completed DNA molecule, double-stranded DNA. Strand here. Here's the nucleotide that I first started with the S-phosphate cytosine here. And I made this in the 5' to 3' direction. Then you switch them around, and I started here to do the base pairing, thymine onto the adenine, synthesized from 5' to 3'. So it's always going, the synthesis is always going to occur 5' to 3', again because of the way the nucleotides are structured. But then notice you have an opposite polarity to the strands. 5' end up here, 3' end down here, 5', 3', and then on this side it's opposite. So these are called anti-parallel strands. 5' end up here, right, 5' carbon, to the 3' end down here. So 5' to 3', 5' to 3' in opposite directions. There's 5' to 3', 5' to 3', opposite directions. Now I know one thing you're saying though, this is double-stranded, but it's not a double helix. It's not twisted. So I can twist this, and if I twist it, then it really does look like a double helix of DNA. And the reason that it twists is because it brings the bases closer together. This is a subtlety of intermolecular forces within the molecule. It would lay like this if it could, but there's forces of attraction between these base pairs. It's called stacking. So what happens is, if you notice, when I twist this, the base pairs are brought closer together. They attract one another because of some force of attraction. That's what induces the twist in the molecule. That's a double helix of DNA. Now RNA can take the same kind of a structure, but you have to be wary about what kind of genetic material you're describing. In cells, any kind of cell, prokaryotic, eukaryotic, doesn't matter. In cells, DNA will always be double-stranded, and it will always be that helical type of structure. In cells, only in cells, RNA is always single-stranded. It can loop back in itself, it can form some base pairing and so forth, but it's always single-stranded. In viruses, all possible combinations happen. Some viruses use DNA as the genetic material. Some viruses will use double-stranded DNA. Some viruses use single-stranded DNA. On the other hand, there are also RNA viruses. RNA viruses will use double-stranded RNA, as in the coronavirus. The coronavirus is called a double-stranded RNA virus because it uses double-stranded RNA as its genetic material, not DNA. There are also single-stranded RNA viruses. The Ebola virus is a single-stranded RNA virus. And those have just been in the news, obviously, because they're like causing havoc in the human world. The Ebola virus has faded away because of immunity and the restrictions that were happening in Africa at the time. But now everyone's worried about the coronavirus because it's spreading so rapidly and has such deleterious effects on people. In any case, that's a double-stranded RNA virus. Same kind of layout as DNA. It just happens to be you get a different sugar in there, you get one of the different bases. You get a uracil in place of the thymine that's going to pair up with the adenine. But here is double-stranded polynucleotide chains. DNA in this case. That's it for DNA.